Hi everyone, Brenda Gustin here, and I'm in the second lecture of microbial genetics. I am here in the lecture study guide instead of the book. Like I said in the previous video, the book has all the information you'd ever want to know about this, but in order for me to just tell you about some of the differences between bacterial cells and eukaryotic cells, um, I don't need you to concentrate on all those details. I just need you to have kind of the big picture. So our first learning outcome says provide an overview of the flow of genetic information and the relationship among DNA, RNA, and proteins. Now, this is something you've learned in other classes. I'm quite sure you probably had it in high school, and you've probably had it in another college biology class. So you have a basic understanding of this, and you want to kind of recall some of that information. Before we go any further, though, I want you to see this term flow of genetic information. If I say that or if somebody says that, they're typically talking about how DNA is used to make RNA and RNA is used to make proteins. And remember, proteins are the molecules in our cells that perform the functions. They perform the structure, they're the structural molecules, they're the enzymes. And so it's really important that our cells make enzymes. Our cells make proteins, and DNA is the instructions on how to make those proteins. And so here we call that the flow of genetic information. You might have heard that called other things. In fact, one of the adaptive learning questions asks you about, they call it the central theme of molecular biology. And they're talking about DNA is used to make RNA, which is used to make proteins. The central theme of molecular biology. I've also heard that called the central dogma. And so this is a really important concept to biology and to how cells function. So don't get kind of confused about what they're asking for. But this flow of genetic information, the central theme of molecular biology, the central dogma, all of that is just asking you that DNA is made from RNA, which is used, or I'm sorry, DNA is used to make RNA, which is used to make proteins. Now, you probably also have some memory of the concept of transcription and translation. And so those two ideas together are just maybe what you want to review, or if you're feeling pretty good about it, just move forward. If you're a little rusty on your flow of genetic information, I put a link here to a Khan Academy video, which I thought was a good one. There are lots of different videos you can use on the internet that describe the flow of genetic information. So if you're rusty, review it. You really, it's an important concept, not just in biology, but in microbiology, in healthcare. It's something that is important to have some concept of. So let's see. So when we're looking at that flow of genetic information, what I did is I put a diagram. I'm going to skip this first one. I put a diagram in the notes, and it just is a very simplistic diagram compared to what they have going on in the book. But here's our flow of genetic information. Remember, DNA, that's our instructions on how to make proteins. DNA is a nucleic acid. DNA makes up our chromosomes, whether you're a bacterium or you're a human cell. And so DNA, when cells divide, DNA is replicated, so it makes copies of it for new cells. But here's the flow of genetic information. When DNA is used to make RNA, we call that transcription. DNA sequence is copied, actually a complementary DNA sequence is copied to make an RNA molecule. And then that RNA molecule is translated, and hopefully you remember that ribosomes are the site of protein synthesis. So this happens, our RNA attaches to ribosomes, translation occurs, and the RNA sequence is used to determine a protein sequence. And every protein is going to be unique, or every type of protein will be unique because of their unique amino acid sequence. That amino acid sequence is determined by the sequence of the RNA. Now, when we're looking at this flow of genetic information, the DNA is, usually one gene at a time will be transcribed. And so if you have a bacterial chromosome, a whole big circular chromosome, just one of those genes, remember we said in the previous video, E. coli has about 4,000 genes. So just one of those genes on that chromosome, 1/4,000th of it, will be transcribed to a piece of RNA. So the piece of RNA that's made from DNA, that's copied from the DNA, is going to be much smaller than that whole chromosome. And then there'd be enough information in that RNA to make just one protein. So through translation. Now the idea is the chromosome, the DNA is going to stay the same in the cell forever, and or almost the same, and the RNA is going to be made as you need new proteins made. You don't need all your proteins in all your cells made all the time. You only need certain ones. My skin cells have genes on how to make liver cells, but my skin cells don't need to make liver cell proteins. So those genes aren't being copied. They're not being, I don't have liver proteins in my skin cells. Okay. So not all proteins are being made all the time. And so DNA molecules are going to stay in your cell all the time, or in the bacterial cell. The RNAs are going to be copied. They're going to be temporary copies. You're going to make proteins from them. They'll disintegrate after a certain period of time, and you'll stop making those proteins. Okay. If you need more proteins, you'll make more RNA. So DNA transcription, RNA translation makes proteins. Now I have one crazy thing here, and that's something going backwards, where we have RNA going backwards to DNA. Remember, we have some viruses that don't have a DNA genome, they have an RNA genome. So when they need to make proteins, some of these RNA viruses actually have a unique enzyme called reverse transcriptase, reverse of transcription. So they're going to go from RNA to DNA, back to RNA to proteins. And the reason why I even mention this is because particularly the HIV virus, the virus that causes AIDS, is an RNA virus. And so it has to have this enzyme, reverse transcriptase, where its RNA is copied back into DNA. And then the DNA is used to make RNA and proteins. And the reason why I mention this is because I know in nursing they talk about some of the drugs that are used to inactivate HIV virus. Some of the drugs that people take to squelch that virus or stop that virus from multiplying in their body. And there are different drugs that stop the function of reverse transcriptase. So you could see how that could be very specific for the HIV virus if human cells don't do this, but just that particular viral cell does that. Okay, so there are reverse transcriptase inhibitors. Okay, so let's go back and look at our learning outcomes. So a basic overview of DNA and RNA. I covered reverse transcriptase, and I just wanted to back up and go into just a little bit about structure of DNA and RNA. And you should be familiar with this again from previous courses. Most people are familiar with the idea that DNA is a double helix. It's got it looks like a ladder, but it's twisted. RNA's got a single strand. Hopefully you remember that there's sugar phosphate along the sides of the ladder, sugar phosphate, and then these nitrogenous bases make up the rungs of the ladder, the steps. And you even might remember that RNA has a little bit different base composition than DNA. But the thing that I want you to notice that you might not have noticed before is that these sugars. So we said the sides of the ladder of the DNA molecule has deoxyribose. It has sugar phosphate. RNA has ribose, sugar phosphate. So ribose is a sugar. Ribose and deoxyribose are really similar, except you can tell by the name that deoxyribose is missing an oxygen. So if you look right down here, oops, there we go. If you look right here, that hydrogen right there, or that carbon, doesn't have an oxygen on it. There's an oxygen right here, but not here. Deoxyribose. And for a reason that chemists and physicists understand better than I do, that makes ribose less chemically stable than deoxyribose. Missing that oxygen in the ribose makes RNA less chemically stable than DNA. So if we go back and we look at our flow of genetic information, whereas we said DNA it makes up the chromosomes and they're always in the cell. And the RNA is made as needed. If you need these certain proteins, you're going to make up some RNA, and the RNA can come and go. So that kind of makes sense that DNA would be more chemically stable than RNA. We're going to talk about another example of why that's important in the microbial world in a future video.